first of all, a warm welcome to everyone present on this meet. Today, we are going to have another very interesting talk on the antimicrobial resistance or AMR, which is again a very burning topic of today's scientific world. So, for that, we have our speakers of today is Dr. Atul Tiwari. So, before I introduce him, just a brief introduction about this Science Dialogue series. So it is an initiative by Dr. Gopalji sir. Uh, 1.5 years already being, uh, uh, it's been already 1.5 year since the beginning of this series. So in that duration we have multiple speakers from the different backgrounds with different expertise, and all of them they have helped us to carry out these free science dialogue series lectures so that the knowledge can be shared to each and everyone who are joining us in this science dialogue series. So we are thankful to all of them. And talking about today's session, so today's session it is majorly focusing on the tailoring of this new advancement in the antimicrobial resistance. So our today's speaker, he is Dr. Atul Kumar Tiwari, who has obtained his Ph.D. from the Department of Chemistry at IIT BHU Varanasi. He completed his master's in microbiology from Dr. Ram Manohar Lohia, Awadh University, Ayodhya, with over 6 years of research experience in nanobiotechnology, nanomaterial chemistry, and biosensor development. He has published more than 25 research articles in very different esteemed international journals, and his research primarily focuses on developing advanced antimicrobial nanomaterials, drug delivery, and the development of the biosensors that can be suitable for detecting the different antimicrobial resistance. So, sir, I welcome you to this platform and hand over this virtual meet to you so that our participant can learn from you, can get benefited from you. So, uh, sir, we can start you.
Okay, ma'am, thank you for your kind intro. I am Dr. Atul Tiwari. So I am just going to give some perspective about the current scenario, world scenario of antimicrobial drug resistance. What are the main microbes that is listed by different health agencies across the world as a common threat for the world health and patients also. So I will not go in very much detail because you are very aware about everything, different strategies that are many researchers across the globe is opting to tackle these kind of drug resistant challenge. So I will just provide a perspective to just put your point of view and acknowledge the what is the current challenges in current world scenario about the AMR. So before going in deep, so I'm just showing an interesting car here. You can see that the tires are very damaged. So what you guess about this car which have tires are damaged? So above the part of this car, the white color bonnet and all these things, whole car is our medical burden on the uh, globally renowned hospitals and clinicians, and the tire is the antibiotics that is bearing the load of that. But why these tires are damaged? Because most of the antibiotics in to date are drug resistant. So next, so just a very simple thing that I just want to emphasize the what is the current scenario of AMR and what about the antibiotics in to date. So this is the another thing. The microbe is saying I am strategically safe. Because these microbes, drug resistant microbe, have different kind of very molecular systems that inactivate the antibiotics in many ways. So what is the antimicrobial resistance? First question arises. So antimicrobial resistance happens when germs like bacteria and fungi develop the ability to defeat the drugs designed to kill them. Because not all microbes are harmful to us. Most of the microbes are also beneficial, but there is also a drawback of antibiotics in this scenario. They also kill some beneficial microbes as well. So antibiotics most commonly uh, very targeted towards a molecular target inside the cell. So but with the evolution and progress of time, the bacteria and fungi get resistant to these drugs due to some very common causes that I will discuss in next slide. That means the germs are not killed and continue to grow. Resistant infections can be difficult and sometimes impossible to treat. So here is some data that is saying about the antimicrobial drug resistance burden globally. Uh, it is produced by CDC in 2019. That saying is around to 1.27 million people killed in uh, 2019 and associated with 5 million deaths. So another thing is 4.6, what is the cost of these medical burden on global economy. Around 4.6 billion annual cost of six type of antibiotic resistance claimed annually. uh, so it is expected that globally around 10 million deaths are expected by 2050. That will cost a huge money and to the global economy. Next, so the question arises, when we take antibiotics from a practitioner or from some local clinicians, after taking antibiotics, how bacteria or fungus acquire drug resistance? Because since the antibiotics are formulated and developed to very targeted killing to against a particular microorganism or pathogenic bacteria. So there is two means that some microbes acquire drug resistance. First is natural mean and second is acquired mean. So naturally, normal cells just grow, in fact, to living organism like humans. But after uh, pressurizing with antibiotics or any kind of small molecule that have the inhibitory action against that pathogenic microorganism, these bacteria sense and just protect themselves from action of that particular drug. So in this way, overuse of antibiotic and misuse in the means that sometimes we take antibiotics even in the common cold, that is not mostly associated with the bacteria or fungus. It's associated with viral infection, common cold, but we take antibiotics. So sometimes the bacterial microbiome system inside our gut, which are not pathogenic, they get pressurized with that antibiotics and can convert or evolve themselves to defend that antibiotics. So naturally, over the time, in a evolutionary process, these bacteria or fungus modify the target site of that particular antibiotic and protects themselves from the pressure of the drug. And after acquiring the resistance against a particular antibiotics, they can also transfer their genomes, because the resistance develops in genomic system in their DNA or genome. So they can transfer that genome, their genetic material to other bacteria and other bacterial strains or species. Why? By a mean of conjugation and different transduction and different type of which have a uh, their uh, generational process. But sometimes, in the other scenario, sometimes these bacteria spread in environment and transfer resistantly to other competent cells and convert them to resistant cells. So there is two means of evolving the antibiotic drug resistance among the microorganism. One is natural and second is environment. So molecularly, at the molecular level, these bacteria, drug resistant bacteria, play very smartly. Sometimes they modify their porine system. Porine is especially for the transportation of water from inside to outside the cell and make the balance of water and several other micronutrients. And also, when we put the antibiotic in the system, some antibiotics enters the inside the cell through via the pore system, porine transporters. So bacteria or fungus play very smartly and just modify their they modify the selectivity of porine towards a specific antibiotic or drug. So drug remain outside the cell. Second, production of some enzymes that cleave the antibiotics into different fragments and inactivate them. Third and most common and most important is the overexpression of a flux pump. If somehow any antibiotic get entered in the cytoplasm of that particular bacteria or fungus, the pathogens just efflux that antibiotics by using their efflux pumps without any delay. Third and fourth system is sometimes, depending on the species of bacteria or fungus, they can, and also depending on the type of drug, they can also modify the intracellular target sites like ribosomal modification, because some antibiotics act on synthesis and assembly of ribosome and also interfere in the protein synthesis. So they modify the target site in the ribosome, and some other ways can be the mutation in DNA guidage and topoisomerase systems. So these are the some molecular basic concept about the evolvedness and molecular concept of how pathogenic drug resistant pathogenic microorganism inactivate the action of a particular drug. So most important and most dangerous form of microbes that are very threatful is the typical bacterial biofilm. Once bacteria or fungus harms the biofilm, it is very hard to remove or kill the bacteria inside the biofilm. So current date challenge in nosocomial infections are bacterial biofilms, because in ICU system, medical devices or catheters are usually used for to relieve the patient, but most of the time bacteria or fungus harms the biofilm or that catheter or medical device and protect get protected by extracellular material from the outside pressure of antibiotics, because antibiotics are unable to evade the extracellular polysaccharide and unable to enter in the microenvironment, biofilm microenvironment, because the presence of some specific enzymes and specific proteins that cleaves or binds with the particular antibiotic and inactivate them. So moving forward, since 1930, penicillin discovered, several antibiotics, either they are synthetic, natural, or semi-synthetic, lots of antibiotics developed over time, and but unfortunately, in 1960s, methicillin-resistant Staphylococcus first observed. So after around more than 50 years, MRSA is a bigger challenge at. So, but we have no option to treat or these kind of drug resistance bacteria or fungus. Fungus, because I am saying fungus is most common drug resistant fungus is Candida auris and some other species of Candida. So, but and unfortunately, you have also observed some the upsurge of mucormycosis in the duration of COVID-19. Uh, uh, Very high cases and mortality rate observed in the COVID-19 patients due to the infection of mucormycosis that was caused by the some specific species of rhizopus like Rhizopus eryzus and Aspergillus species, some in some cases Aspergillus species, and in some cases the Rhizopus homothalicus, that is very rare fungal. So what we have opted, because amphotericin B is used as a frontline antibiotic against these kind of fungus, but unfortunately, the solo amphotericin B without any combination was mostly ineffective. So you have, I think you have learned or listened about the liposomal amphotericin B. So why we use liposomal amphotericin B? Because liposomal, because liposome just directly deliver the amphotericin B inside the cytoplasm and increase the bioavailability of amphotericin B. So here you can see the lot of antibiotics get developed over the time, but here is the main scenario. Some significant drug resistance pathogens are the current challenge, like carbapenem resistant enterobacterials, Staphylococcus aureus, Enterococcus, Acinetobacter bomini. Acinetobacter bomini is generally the main cause of catheter and medical device associated bacterial biofilm, and is most of the antibiotics, around 80% of antibiotics known current date, are ineffective against the biofilm of Acinetobacter bomini. And multi-drug resistant Pseudomonas aeruginosa, Enterococcus, Helicobacter pylori, all these microbes are mostly drug resistant, as data generated by CDC and WHO in 2024. So overall, after seeing this scenario, we can say that our health system and the life of patients is in danger due to the increasing evolution of antimicrobial drug resistant microbes. So the next question is, if most of the antibiotics are drug resistant against a particular pathogen, then what is next? Why we are not developing some new antibiotics to tackle the challenge? So the fundamental challenge associated with the development of new antibiotic is to identify and develop novel antibiotic that can withstand with the rapid onset of existing antimicrobial resistance is a daunting task. The creation of a new pharmaceutical is a multifaceted, highly expensive and time-intensive process. So I hope you learned about the development and pharmaceutical formulation of COVID-19 vaccine was covered in a very short range of time. But many opposite questions are also raised during the time that how a vaccine can be developed in 6 months or 1 year period. So there was also a controversy on the development of vaccine in scientific community, but somehow vaccine work well, and we are get benefited from the vaccine. So like that, the development of antibiotic, a single antibiotic, either discovery of new antibiotic by natural mean, or now the in the era of artificial intelligence, most very renowned group of researchers across the world is developing and discovering the novel targets, and screening the different kind of small and large molecules for the uh, development of novel antibiotics, and also repurposing the drugs, pre-existing drugs, to create a hybrid antibiotic that can be useful to tackle the challenge of AMR. So if the discovery and development of a novel antibiotic is time-taking and daunting task. So, what is the way forward? So, it could be combination therapy. Why combination therapy? And currently, most of the countries and most of the renowned hospitals in the world, including India, they using combination therapy, because combination therapy makes antibiotics multi-target and broad spectrum. Suppose if we are taking posaconazole and amphotericin B in combination, they uh, uh, the combination of amphotericin B and posaconazole against the rhizopus species, they limit the germination and inhibit the germination of sporangiosperm of rhizopus. Uh, and one of the hospital at I think IMS BHU, where I worked during the COVID-19 time, on the eradication and some testing of combination therapy against Rhizopus homothelicus, we found that combination therapy was very responding very well. When we tested the amphotericin B and posaconazole in alone. So first focus should be on combination therapy. Second, novel drug delivery systems, because the pathogenic fungus or bacteria uh, just pump the antibiotics from cytoplasm to extracellular environment. Uh, so, what could be next? Suppose if we retain that antibiotics in the cytoplasm for longer time, and uh, bacteria or fungus or pathogenic microorganism, any can be not able to efflux that drug outside the cell. So it would be more reasonable that it will bind to their target. And third could be the alternative molecule and advanced materials, because some chemistry, due to the advancement in chemistry, synthetic chemistry, and involvement of artificial intelligence, advanced material can also be used as an antibiotic. And fourth and most challenging task is development of multi-target broad spectrum antibiotics. But during meantime, if development of new antibiotic is taking time, too much time and expensive, so we can use the first two options like combination therapy and novel drug delivery system. So in this figure here, you can see in this figure different kind of materials and options are available to tackle the antimicrobial drug resistance. Some of the inhibitors are also getting discovered for different kind of enzymes, against different kind of enzymes, efflux pumps, and many other cellular processes. Antimicrobial peptides are also very interesting, but taking much time due to the cytotoxicity and non-selectivity. Antibiotics and other thing, self-assembled peptides, polymer-based antibiotics, biomimetic nano delivery system, carbon-based nanostructure and liposomal materials, photosensitive materials and cationic polymers, metal nanoparticles. So there are a lot of alternative option that could be the a new weapon to tackle the antimicrobial drug resistance. But the question is how much time, how much effort, and how much suitability it would require to achieve that task. So I am putting some current scenario, that what kind of drugs or antibiotics are in pipeline uh, across the world to new antibiotics. So some of the drugs are in phase 3 clinical trial, that is last stage development, like azithromycin, avibactam, developing by Pfizer, and Abbvie, joliflodacin, Inovia, and GRDP, developing by GRDP, Jepto, T-dicin, GlaxoSmithKline, GSK is also developing and in phase 3 clinical trial. Josura Balpin also developed, getting trial by the Roche and Harvard University. So a lot of antibiotics are that are in pipeline and in the way to come in the market if everything the will be in favor of the patient and human physiology. And some drugs are also in different phases, like phase 1 and phase 2 clinical trials. However, the phase 1 and phase 2 clinical trials are very early stage. We cannot say much about the suitability and approval of these drugs. So by the way, but we are some research group across the world is working on these uh, antibiotics or antimicrobial drugs. Hope so they will come to the market. So recently approved and newly launched antibiotics are mostly 2 or 3, like Zenis, Workhard, and Byrec. It is planned to launch in India in 2026. It targets the multi-drug resistant gram-negative infection. However, the still gram-negative, gram-positive bacterial strains are in consideration, and fidaxomicin, yeah, developed by Merck, is a microcyclic antibiotic approved from Clostridium difficile infections, superior in lowering recurrence rate versus vancomycin, because it is fidaxomicin is superior, have superior activity as compared to vancomycin. So because my expertise in especially in drug delivery, so I will not talk much about the other approaches and alternative options to tackle the AMR. So I will mostly focus on some my recent work about the drug delivery against to tackle the antimicrobial drug resistance using some nanoparticles. So here are some different kind of nanoparticles that are useful to deliver the traditionally and pre-existing antibiotics like vancomycin, penicillin. However, penicillin is no more in the movie, but some workers across the world is working to revive the penicillin, because penicillin was the first antibiotic that discovered and utilized very well to save the humanity. So inorganic metallic nanoparticles are useful, that can deliver some pre-existing and traditional antibiotics. Polymeric nanoparticles, liposomes, nanoemulsions, NLCs, SLNs, and these kind of nanoparticle, mesoporous silica nanoparticle, and biomimetic nanoparticles are also can be synthesized and load the particular antibiotic to deliver inside the cytoplasma pathogen, specific pathogen. And these nanoparticles have ability to retain that drug inside the cytoplasm, and makes the higher possibility that the delivered antibiotic get interact with their target site and inhibit the growth and proliferation of a specific pathogen. Some of the antibiotics pre-existing, as the title of this talk is the traditional antibiotics. So some of the nanoparticle based drug delivery systems were tested over the period of time, uh, using the traditional antibiotics like polymyxin B, methicillin, oxacillin, gentamicin, colistin, ciprofloxacin, and many more antibiotics have been studied under in vitro and in vivo condition to tackle the AMR and eradicate the pathogenic microorganism. However, till date, uh, we are unable to deliver any uh, such antibiotic that is uh, effective via nanoparticle drug delivery system, except the liposomal amphotericin B that is available in the market, and very crucially and used for the terminally ill patients who was infected with the COVID-19 associated mucormycosis. So some of my work that is associated with this talk is I'm going to present here. So I used vancomycin, that is a glycopeptide antibiotics. So vancomycin usually used for the gram treatment of gram-positive bacterial infection like Staphylococcus aureus, against the methicillin-resistant Staphylococcus aureus, but it is usually inactive against gram-negative bacterial infection. Why? The question is why vancomycin is inactive against the gram-negative bacterial infection? Because vancomycin binds and stops the transpeptidation in the bacterial system that forms the peptidoglycan layer around the cell, and vancomycin just stop the transpeptidation reaction to form a network of NAM and NAG uh, uh, polysaccharide around the cell, the network of NAM and NAG. So if that NAM and NAG transpeptidation inhibited, the peptidoglycan layer get very fragile and could not retain the architecture of bacteria. So major challenge in treating gram-negative bacterial infection with vancomycin is the presence of outer membrane layer that is just above the peptidoglycan layer. So that outer membrane layer protects the cell from entering the vancomycin and limit the accessibility of vancomycin to the peptidoglycan layer. So how we can make it active? So the question arises that how we can make the vancomycin active against such kind of gram-negative bacterial infections. So Here you can see the differences between the bacteria gram positive, gram negative, and fungal uh, cell wall structures. So as I said that it vancomycin just inhibit the glycosylation or peptidation reaction, transpeptidation, and inhibit the complete formation of peptidoglycan layer. So I used to synthesize some gold nanoparticles, which was just conjugated with vancomycin without changing their intrinsic property. Because vancomycin is glycopeptide, so we found that the the vancomycin very well get conjugated very efficiently with high loading capacity on gold nanoparticles. So and found that the conjugation was in very effective, and it was very rational. And before that, no one around the world reported the interaction of vancomycin with the gold nanoparticle. However, some reports were available to the interaction of vancomycin with copper nanoparticles, but gold nanoparticles was not reported before that. So we developed a system that conjugated gold nanoparticles and applied against some here. Here you can see the developed nanoparticle. Here the layer of vancomycin you can see on the surface of nanoparticle. Uh, and here is the some chemical reaction that how the conjugation and all these things uh, get happened. So I am moving forward, not going in very deep chemistry. So here you can see, because vancomycin is a glycopeptide. So intrinsically vancomycin is itself a fluorescent molecule. So we can and most lot of work had been done to differentiate between the gram positive and gram negative bacteria, and diagnosis using the vancomycin intrinsic fluorescence. So here you can see that after conjugation of vancomycin with gold nanoparticles, the fluorescence of nanoparticle get significantly enhanced. Here you can see in that the spectrum, and here is the you can see the color of nanoparticle after the conjugation, emission range of nanoparticle after the conjugation of vancomycin. So we thought that we can localize the vancomycin loaded conjugated gold nanoparticles around the bacterial and fungal cell. However, vancomycin is not active against fungus. It is specifically active against bacterial gram-negative bacteria like Staphylococcus aureus. But we used as a just our curiosity, and found that even Candida albicans, Candida tropicalis, and some gram-negative bacteria like E. coli and Pseudomonas were also get uh, attached with the developed vancomycin conjugated gold nanoparticle. Here you can see the blue fluorescence, blue dots. That the significant that is especially showing the presence of vancomycin conjugated gold nanoparticles around or inside the cell. And here you can see this is the cell of Candida albicans. Here you can see that the nanoparticles are filled inside the cell. Here is more detailed overview that how this is the blue channel that indicating the presence of nanoparticles around or inside the cell, and here is the red channel that is cells where stained with propydium iodide that obviously show the viability of the cell. So you can see that having nanoparticle filled cells were almost dead. So we again estimated the dead percentage. Here you can see the Candida albicans was somewhat showing less sensitivity because vancomycin is already not active, not against the Candida albicans, and so very less mortality. Dead percentage was 16.3%. However, interestingly, that Candida tropicalis were showing a huge dead population. Again, E. coli was not very sensitive, 12% dead. Pseudomonas aeruginosa again shown a very huge dead population. So overall, we can conclude that the developed nanoparticle, vancomycin conjugated nanoparticle, the activity of that system was species and strain dependent, depending on the variability in the cell structure. Here is the some data that that population and generation of reactive oxygen species that is also aligned with the dead population. Yes, here you can see the term of the treated microbial strains. This plate is the control Candida albicans. Here is the you can see that treated Candida albicans have some pore and pore-like features with cracked surface. But in case of Candida tropicalis, here the whole cellular architecture was collapsed. Again, in the case of E. coli, the nanoparticles were attached to a specific location uh, on the cell that indicated the site specific interaction of the developed nanoparticle vancomycin conjugated system. Again, in Pseudomonas aeruginosa, here you can see that some of the nanoparticles were attached to the surface of the Pseudomonas cell, but not on overall bacterial surface. So we can conclude that there is some targets, some of the specific molecule on the bacterial surface that was get interacted first with the nanoparticle system, and further the cell was not collapsed, it was intact. So here in this slide, you can see the graphical representation of whole work of how we developed the vancomycin conjugated gold nanoparticles and their impact on different broad spectrum microbial against the broad spectrum microbes, even its candida and gram negative bacteria. So overall, we can say that the delivery of traditional antibiotics against uh, the pathogenic microorganism that is resistance in a way that just effluxes the drug outside the cell is very interesting and can be used and explored for further advances in the tackling of AMR. So a lot of possibilities still we have to focus on such kind of strategies to retain the traditional antibiotics in mainstream uh, uh, clinical system, along with development of new antibiotics. So here we are working some small molecule that is very site specific, and here you can see that these small molecules are very significant, have broad spectrum antimicrobial activity, and they are localizing in certain organelle specific manner against the gram positive, gram negative, and again also against the fungal cells. So this image is also indicating that there is a lot of chance that we can develop some new drug delivery system to tackle the AMR. So thank you everyone for your attention.
Okay, thank you, Atul sir, for delivering such a nice talk and giving us a kind of perspective for the delivery of this vancomycin against the both gram-negative bacteria and how this resistance can work. So now I would like to ask the participants if they have any queries. They can directly ask, raise their hand, introduce themselves, and can have the interaction with Atul sir.
Yes, please.
Uvesh Patel, please unmute yourself.
Uh, Good evening, sir. I'm
Yeah.
Umesh Patel from University of Salford. Currently finished my Masters of Biotechnology. I have done the similar research like you on nanoparticles to tackle antimicrobial resistance. So I have used the amoxicillin and tobramycin with conjugation of cuprous oxide nanoparticles. So my question is, I have found a similar thing and sensitivity, but I didn't calculate the concentration of nanoparticles. So
Yeah.
I want to ask you that have you calculated the concentration of nanoparticles by NTA, and have you used the ROS confirmation test like propydium iodide test?
So generally, you are asking that how can we estimate the concentration of nanoparticle, or what is the exact what are the exact question?
Yeah, that is my question. That have you? I like
Yeah,
I'm
it's very
asking the
easy.
question.
It's very easy. You can quantify the concentration of nanoparticle and also the drug locked in capacity of that nanoparticle. It's very commonly available in literature. I suggest you go through the Google and type your question. You will definitely find the answer. If you have any further challenge in the quantifying these kind of things, then you can contact me via email. I will definitely help you with that.
Okay, okay, thank you, sir.
Okay, anyone else? Uh, okay, so I think that there are no more questions. So, sir, thank you very much on
Thank you,
behalf
ma'am.
of
Thank you. Thank you
on.
for providing such an opportunity to just
I
help.
actually the credit merely goes to Gopalji sir for this kind of uh, what I can say. This kind of invitation to different speakers of the
It's
experts.
okay, ma'am. I think Gopal sir is busy somewhere because I am familiar with this platform because it's my second last talk today. Today is my
Okay,
second
madam.
talk. Before that, I just
Okay.
talked about some biosensor development programs.
Okay, sir.
So
So, sir is actually busy in some
yes, yes,
pre
yes. I
assignment.
can understand.
Uh, thank you very much for joining us in this platform and delivering
Okay.
such a nice talk. And hopefully, we will be having some more lectures in near future also.
Yeah,
And
sure,
thank you
sure.
all the participants for joining us in this lecture series. And we hope to have more lectures in the coming days. So we'll be looking forward for your participation in those lectures also. So thank you everyone. I would like to now officially close this meet. So sir, thank you so much again on behalf.
Thank you, ma'am. Thank you, ma'am.
Okay, thank you everyone. So we are now closing this meet.